Hi, my name is Li Xin Chen. I'm a research scientist in the DNA enzyme division here at NEB and a member of the Prevalence lab. Our recent publication in the journal Science is titled DNA Damage is a Pervasive Cause of Sequencing Errors, directly confounding variant identification, and was performed as a collaboration with Lawrence Aquilar, a staff scientist, and Pingfang Liu, a development scientist here at NEB.
The study has major implications for the high-throughput sequencing technologies and the detection of somatic variants. Somatic variants are sequences that are unique or share with only a few cells in our body. If somatic variants alter key genes responsible for the control of the cell proliferation, they result in cancer. During the growth, cancer cells acquire additional somatic variants, leading to populations or subpopulation of cancer cells and tumor heterogeneity. The subpopulations of cells can be resistant to otherwise working therapy and are therefore responsible for the relapse of the cancer. Thus, it is important to accurately identify all somatic mutations, even the ones that are specific to only a small subpopulations of cells.
The development of high-throughput sequencing technology has revolutionized our understanding of cancer biology and the treatment of cancer. Sequencing of different tumor samples has identified many previously unknown somatic variants, many of which have been successfully applied in targeted cancer therapy. In addition, the ability to perform large-scale sequencing of different tumor samples has enhanced our view of tumor heterogeneity. We now have a much better understanding of clonal selection and expansion, drug resistance, and individual variation in response to targeted therapy. The continued success in applying next-generation sequencing to cancer therapy will largely depend on the accurate identification of somatic variants of high sensitivity and high specificity.
Sequencing accuracy directly impacts the identification of somatic variants. Not surprisingly, sequencing chemistry is not perfect, and every so often, errors occur. These errors were usually ascribed to amplification mistakes or sequencing chemistry. With modern high-accuracy polymerases and sequencing chemistry, other sources of error can become problematic. What makes this study so important is the discovery that DNA damage can be a prevalent source of sequencing errors, and fixing these damages greatly improves the detection of true somatic variants. Not all damage leads to sequencing errors. Some lead to a loss of signal, while others are more insidious, causing a polymerase to read the damaged base differently than the undamaged base. Guanine bases are known to react with oxygen, forming 8-oxoguanine, which is read by many polymerases as a T instead of a G. This is insidious because, unlike strand breaks, where no information is generated, you instead get incorrect data, which can look like a rare somatic variant.
In this work, we identify DNA damage as a prevalent source of sequencing errors by using two independent strategies. The first strategy uses a DNA repair mix, such as pre-CR or the FFPE DNA repair mix, to repair the damage present in the DNA sample. By sequencing the same sample with and without action of DNA repair, we can quantify the level of damage present in a DNA sample.
The second strategy uses the fact that the damage triggers the polymerase to always respond with a similar behavior. For example, as Tom mentioned, 8-oxoguanine is misread by the polymerase as thymine instead of guanine. Sequencing of 8-oxoguanine containing DNA will result in an imbalance between the rate of G to T compared to the rate of C to A variant, which is the reverse complement of the G to T. The difference between these two rates corresponds to damage. Based on this principle, we computed a simple metric called the GIV score to measure all damage in the DNA and validated this matrix using the experimental setup described by Li Xin. The next question that we wanted to address now is how prevalent is this damage in sequencing facility that handle real cancer samples. We therefore requested access to the Cancer Genome Atlas Initiative data and found that more than 80% of the dataset have a significant imbalance of G to T, indicative of damage. It is likely that the majority of sequencing currently done have large degree of undetected damages. Consequently, the rare and possibly interesting variants are confounded by incorrect data derived from this damage.
For more details about this work, You can read our publication in Science in the February 17th issue, 2017. This work is also available in open access on bioRxiv.